Now, before we go to the poster session, we wanted to have a kind of pitch session. So all the posters and presentation, you have 1 minute, because we are 30, you are 30 people. So you have 1 minute to pitch your poster. So no slides, just you have to convince why everyone should pop up around your posters. You have the numbers, the demo. I think you don't have numbers downstairs, so make note of your number, because there is a best poster award possibility. So you can vote in that QR, and you will have the best poster or demo will have a prize. So you will have, I will set up the timer. You will have one minute. So you see the order when you have to present. So starting with the SPARQL Federated Query debugging tool.
Okay, SPARQL Federated debugging tool. SPARQL Federated Query, we are talking about the SPARQL language extension, so about service patterns that can be nested, so they create the service pattern tree, and you can route the subqueries to dedicated endpoints. And during the execution, the problem is that instances of these service patterns are created. They created potential huge tree. And without debugging, you just received HTTP response from the root node from this service execution tree. And you have no idea what's going on during the execution. So the debugger is just rendering in real time this service execution tree, and you can see requests, responses, durations, etc., to knowing what's going on. Sent? Okay, thank you.
So I'm part of a large project which spans, I think, about 3 institutions. It's a cellular genomics project, so we have a lot of different cell types. And we're interested in particularly how much ontological information, let's say, about one particular cell type could be translated to a different one. So there's been a lot of talk about ontology matching already in the conference. But we're interested in doing this using a large language model to try and learn from the large language model whether these names or concepts or even the triplets themselves change given a particular context. So that context could be given to the large language model, or it could be via fine-tuning. And we're hoping that the advantage here is that we can update the model with new information as new papers and literature comes out on the topic. It's fairly early days, but yeah, please do come and visit our poster. And I'm happy to talk about any of the mathematics, because there's quite a bit to talk about here. Thank you.
Poster number 3 is about plant science, actually. And we found that germplasm banks are seen as seed supermarkets where people say, I want this specific seeds for this specific species. But we want to convert them into data centers that can perform research and support research from other organizations and enhance collaboration. For that, we created an architecture that uses the FAIR principles to enhance discoverability and interoperability in particular. And ask me, please, about our query matching system that matches SPARQL queries with endpoints that are responsive to them. And also about how I modify the triple pattern fragments resolution algorithm so that it can be used in a federated system. And that's it.
Hello, so I'm presenting Omexcavator, which is a tool to export bioimaging data to put it in a wider knowledge graph context. So we are having this Omero, which is in fact a bioimaging repository with pretty nicely defined data model and also semantically interoperable. But unfortunately, these metadata records for the bioimages, they rarely do leave the bioimaging world. And this Excavator tool, it creates a JSON-LD interoperable representation for the bioimaging metadata to be put in a wider knowledge graph so it connects to the other research assets. In this, we are also very interested and looking forward to hear about your use cases. So maybe you would like to connect bioimaging data with, say, genetic information or clinical phenotypes. In this case, or if you're also just interested about technical details, see you at poster 4 and very happy to chat. Thank you.
Hello, everyone. I'm Elias. I'm from Ghent University. And my poster number 6, Semantifying Genomic Variant Data, is centered around representing VCF data or variant data as RDF. As we were shown earlier today in the semantic beacons, there isn't really an established schema for representing genomic data as VCF. And so I proposed something here, and I've already talked to the authors of the semantic beacons, and we are looking to I'm looking to start the conversation and talk to people about how we can make a schema that is usable by more than just one research group at a time. So if you'd like to discuss that, come and see me at number 6.
Hi, everyone. I'm Katerina Liberidou, and I'm presenting the DatoSCAT project. So here we're presenting actually our GCAT database, that is a database full of Catalan health data from actually 20,000 patients. And we are creating on top, we do the transformation with the OntoBridge, like an application that is made from Hospital Clinic. And this is the first time that we are using this application outside of the Hospital Clinic. And on top of that, we create the OMO beacon so we can make all the genomic data available for the public and the scientists. If you're interested for more information, you can find us in the poster number 7, and I will be happy to answer to your questions. Thank you.
Hi, my name is Ron. I'm here on behalf of my PhD student, Lea Gütebier, who actually did the work and did the poster. We created a study graph that combines study protocols from clinicaltrials.gov and mostly questionnaires and medical forms from the Medical Data Models portal. And we combined that using the MeSH and UMLS ontology. And we are now able to do fancy Cypher queries querying our Neo4j database. If you want to know more, come to my poster. I also have a demonstration there so that you can see it's real and not fake. Thank you very much and see you later.
Yeah, hello everyone. My name is Julia and I'm representing DSMZ Digital Diversity. We do have a very large portfolio of different databases on the life sciences, especially on microbes, as we specialize in microbes. So you may know some of them, like the Brenda enzyme database, but also the BacDive database on bacterial diversity. And here I want to present our attempts to RDify our knowledge bases. And so, yeah, I'm happy to chat about integrations, about other data types to integrate, and also about collaboration and best practices. Thank you.
Hello, my name is Yomi. For my own project, it's mostly about clinical remission for those who have IBD, inflammatory bowel disease. So the idea was to build a Bayesian network using experts' ideas, generated ideas that basically for concepts that are used to treat for Bayesian network, and also to try it using the computer and see which can predict remission better for now. So basically that was what I did. Thank you.
Hello everyone. My name is Nupur. I am representing poster number 12, anonymization and ontology. This ontology represents generic anonymization software to achieve GDPR compliant anonymization of health data. And the main idea behind this was to anonymize health data and make it GDPR compliant so that it can be used for clinical research and developing new medical devices. Our ontology has metadata annotation and has knowledge representation of privacy methods, which help us in choosing the best anonymization algorithm for the suitable data set or required use case. Thank you.
Hello everyone. So poster number 13, AgroLD, knowledge graph for the plant sciences. So mostly focused on molecular entities such as gene, proteins, pathways, gene interactions, and QTL-GWAS. The poster will be focused on crops, tropical crops. I will explain you how we design the data schema, the pipelines, and there is also web interfaces that the user can use for Sparkle queries or to visualize data. Thank you, and see you at the poster number 13.
Hi, hello, I'm Shuichi Kawashima from DBCLS Japan, and today I will introduce our work on the RDF portal. And the RDF portal is a platform that provides access to high-quality RDF data for life sciences. We are working on an automatic update system to improve data consistency and efficiency. And our approach is to generate void file from RDF data. Then we use our RDF config tool to create model file from the generated void files. So this system is still under development, but in near future, it will ensure RDF model always match real RDF data. And it enables automatic Sparkle generation, Sparkle query generation across databases. And it will also improve data integration and usability. So our goal is to make RDF data more accessible and reliable. Thank you.
Okay, hi, I'm Yasunori Yamamoto from DBCLS. And in this poster, I'm presenting URI synonym problem. So as you all know well, there are many biological concepts, and when you use RDF, we have to use URI for concepts. And also, if we use only one URI for a concept, it's very easy to share knowledge with over the databases. But in real, there are many URIs for a concept. So we are now constructing a dictionary of the URI synonyms. And also, when we use this dictionary, we are targeting not only for prefix differences, but also nomenclature differences. So we want to discuss about these issues, and I want to know what you want to know concerning this problem. Thank you very much.
Hello, so my name is Rick. I'm representing poster number 16. Because there are not enough semantic web people in the room or out there in the real world, we wanted to make it as easy as possible to deploy fair infrastructure, fair technologies. And I will show that in the poster, how we do that. Thank you.
Hi, so in poster 17, we try to improve the machine extensibility in bioinformatics analysis, and we do this with two goals. So the first is to propose a minimum metadata schema for the machine extensibility of tools. And we do this by looking into existing repositories, obtaining their metadata, and comparing this. And second, we also want to align this schema to the metadata of data sets in order to improve their interoperability. So if you're interested in the machine extensibility of tools, or the interoperability between tools en datasets. Kom zien met poster 17.
Hello, I'm Julio from Riken, Japan, and the problem we have in Riken is that we have many RDF data. So we thought that to make use of this RDF data, we could create a AI system that can translate natural language questions into Sparkle queries and leverage this data. But the issue with our data is that there is no example of question and Sparkle queries, so that was an issue, and we don't have an expert to create this curated data. So to solve that problem, we created a framework that automatically creates question and Sparkle queries using common RDF graph patterns and LLM API, which propose the question and Sparkle queries for us synthetically. And yeah, I'm from poster 18. If you want to know more, thank you very much.
Hello, my name is David Steinberg. I'm with Camber Cloud. I'm very excited to be up here today because this is a project that is born from this community. It started in Biohackathon Japan in August and was continued at Biohackathon Europe, Elixir, back in November. The problem that we're trying to address is that biological data is not accessible to the current machine learning tooling. And so we looked at metadata as spec that has been implemented within a couple of different tools and repositories, and compared its usage in our domain and offered some opportunities to improve. So come see us. We're actually in spot number 5. And if you're looking to hack on this problem of machine learning against bioinformatics data sets, we also have a hackathon project. Thank you.
Hi, everyone. My name is Karos Kremers. I'm also promoting a hackathon, Channel 4. So I believe that making RDF shouldn't be difficult for data owners. And I want to achieve that by hiding as much as possible the difficult parts like URIs or data paths or anything else behind a nice UI where we just ask the data owner to drag and drop their data features to the associated class or property of a schema, and then we can take the next step. Often I find data owners not even able to parse any of these concepts yet. So just having this drag and drop and then compare what they said for their first mapping to what we think as data stewards, that's already a big jump. It goes from maybe 20 hours to half an hour, I hope, of work with the data owners. Yeah, so visit 20, it's close to the toilet. So when you're done rushing, you can come back and chill out with me.
Okay. No, hackathon is both the biohackathon, both in Japan, that we have some colleagues here, Toshiaki, Suichi, Yasunori, and the biohackathon Europe, that is usually hosted by the BSC, are very important. The biohackathon in this conference is becoming important as well, so I encourage you to take part in those projects. Last round is for the demos. Okay, so remember to get your number and post it near your panel.
So, hello, my name is Mark from University of Greifswald. I'm representing the LARA software development suite. It's a research data management system. It's designed as a general purpose research data management acquiring system that enables scientists to structure the data without any knowledge of semantic background. So they can just use automation systems that we develop, so automation environments to aggregate all the data, and it's automatically enriched with semantic information and building knowledge graphs that can be then explored by SparkQL as an endpoint. This is done all automatically. That's very important to say. Thanks a lot. Yeah, it's a demo, so I'm at the very right of the demo session, if you would like to join our demonstration. Thanks.
Hi, my name is Francisco Abad from the University of Murcia, and I will presenting together with my colleague Gonzalo CASAR, which is an application for quality assurance of ontologies. Well, the story of this application is in our research group, we have done a lot of work regarding ontology quality assurance, but they were like independent projects, right? So this is a result. So with this application, we try to integrate all of our projects. And well, we are using an ontology to integrate all of them. So we have a quality model, which is expressed in an ontology. And we have a lot of metrics to measure different characteristics of ontologies. And well, we have here in CASAR a very nice application with very nice visualization of the metrics. It's all integrated with GitHub, and well, it's a lot of features that I hope you will like it. So I hope to see you later downstairs. Thanks so much.
Good morning, I am Ornella Herrera and I'm presenting Metatron, which is a web-based collaborative tool for the biomedical literature. Users of Metatron can annotate the documents in several different ways, including entity tagging or linking and relationship annotation. And they can also take advantage of automatic built-in annotations. In addition to this, to keep track of annotators' work and to increase reliability of ground truth collections, Metatron provides statistics and inter-annotator agreement metrics. Metatron is released both as online instance and as Docker container in order to ensure data privacy. If you're interested and you have some questions, I will be happy to answer. Thank you.
Hello, everyone. I'm Enrico Bignotti.
And I'm Aniko Gerencer. We are from the Publications Office of the European Union.
And the title of our demo is Collaborative Management of Semantic Web Datasets, Maintaining EuroCyBOK with Vocbench and ShowVoc. It's a mouthful, but EuroCyBOK is a taxonomy we developed at Cordis, which many of you might know as the website that publishes research projects from the Commission. And it's used to classify the fields of science. And while we develop it, we also need to find a way to maintain it and share it with other users.
And for that, we are using two collaborative tools, which are all open source and using semantic web technologies. Vocbench is a maintenance tool for taxonomists, Zauri, ontologies, and all kinds of RDF resources. As I said, it's collaborative, so multiple users can work at the same time. And it's exploiting linked data features because you can actually link the projects in Vocbench and then have the access to them through the tool. And then ShowVoc, it's another tool from the same family of Vocbench. It's a very user-friendly and easy to browse tool, and it's specifically used for the visualization of the taxonomist Zauri ontologies. It has a nice graph view or tree view, so it has multiple features for the data visualization.
Yeah, so you want to see us, we're at the demo, and you can see how the Commission is doing its best to open up its work.
Hi, me again. I also have a demo next to my poster, if you go downstairs to the right. And I welcome you to try to explore the study graph with me using some Cypher queries. Thanks.
Hey guys, my name is Ilya from Germany, and today I will be sort of telling you the story of our pipeline, which is called the MEDEX Knowledge Graph on Fire, and telling you a little bit about how it works. And basically what it does is takes FHIR formatted clinical data which is the most popular format for storing and sharing data in Germany, and transforms that data into a graph structure, optimizes it, and then integrates it with BioCipher for further ontological enrichment. And yeah, this is going to be the latest version of that pipeline, which we are currently running in our local data center, data integration center in Greifswald. So yeah, if you're interested in clinical data and the various ways it can be visualized and queried and all that sort of stuff, please come see me. Thank you.
Hi again, I'm Pierre Lamont from the University of Montpellier. So for this demo, I will show you the scientific literature-based knowledge graph. So I will show you how we design the pipelines, the NLP pipelines, entity taggers, and how we extract and construct the knowledge graph. I will show you the schema, and then we will have the demo of the web interface that can you can search entities and papers and also browse the graphs for the knowledge graph. The demo will be focused on wheat and rice genomics. Come to see me at the demo number 27. Thanks.
Hi, again, I'm Yasunori Yamamoto from Japan, and I'm presenting the number 29, Glycosmos. Okay, so our demo is to introduce Glycosmos. This is a portal site for database and repositories, and also tools, relevant tools for glycoscientists. So I don't know how many of you know well about the glycosciences, but glycan is very important stuff, and it's a chain of sugars and plays a very important role in living things and organisms. So I'm talking about databases, how can use or how much you can use this database, and also discuss about the future plans. Okay, thank you very much.
Hi, this is James from Sage Bionetworks. So many of our collaborative researchers spend a lot of time organizing data from biomedical PDF papers and Excel files. And this work can be very slow and can lead to mistakes. So our AI agent automatically reads the given JSON LD-RDF files, labels the information, and puts it all into a consistent format. So this saves a lot of time. So it is 90 minutes of manual data curation into a 3-minute AI-assisted annotation, and it reduces errors and help researchers focus on new discoveries. So if you'd like to see how it works, please join us for the demo number 30 for our current prototype. Thank you.
Hello, my name is Frank Meinecke from Leipzig University, and I would present you in the software demonstration the FERDEM-Seek platform we used as local data hub. That's a complete research data information and management platform you can use at your institution to make your work transparent and visible to the outside. It's an easy deployable tool, and we use it for transporting metadata of clinical and epidemiological trials in the form of a metadata schema, and which we developed in the National Initiative for Infrastructures, this NFDI for Health. And see us at number 31. Thank you.
So thank you very much to all the presenters.